I will be presenting on the 2024-2025 tick survey data. So oh, sorry, can you hear me? Yes, I'll shout. So we've heard already about the background of tick-borne pathogens and why they're so important, and the devastating economic losses that they cause farmers. But in order to create new dynamic control strategies, we need to understand the factors that underpin transmission, and these occur across multiple scales. So as we heard before, the microclimate is really important for driving tick distributions. But on a much smaller scale, the microclimate can also alter the tick's microbiome. And that refers to the bacteria, fungi, archaea, viruses, and protozoa that colonize tick living surfaces. And these communities are really important for promoting the health of the tick. For example, they rely on their endosymbionts for to supply vitamin B, which they lack in their hematophagous lifestyles. So the microbiome is really important for the tick. But also there is now evidence that the microbiome members can alter the pathogens that we're finding in the ticks. So they have very intricate relationships with each other. They can be antagonistic or they can be mutualistic. So what we really want to see is whether we can associate certain commensal or symbiotic bacteria with pathogens of veterinary importance and see if there's any patterns there. So that's what I'm going to be focusing on. So already there is evidence to suggest that these relationships do exist. For example, in Ixodes ricinus ticks, the presence of the endosymbiont mitochloria mitochondriae is associated with an increase in the acquisition of the pathogen responsible for Lyme disease. And in dermacentor ticks, their endosymbiont, they've got a rickettsia endosymbiont, and the presence of that is associated with a reduction in the acquisition of a pathogen of known veterinary importance. I've forgotten the name, but we know that these relationships do exist. So it'll be really interesting to see whether we can determine these patterns in the ticks collected and also the role of the environment in shaping these interactions, because it's likely that this will have an effect on the dynamics of pathogen transmission to livestock. So as Hannah covered earlier, our objectives are to characterise the microbiomes of ixodid ticks, so hard ticks of veterinary importance. We want to identify the relationships between the tick's internal microbiome and the pathogens of known veterinary importance. And ultimately, we want to quantify the relationships between the vector microbiome, the environment, and livestock disease, and use this information to help inform future control strategies. So here is a method slide on the fieldwork carried out by Bust and Ori. So there were 100 randomly generated locations across Botswana to make it as unbiased as possible. And the main aim was to collect ticks with blanket dragging, so we can collect the microbiomes of unfed ticks, but opportunistic sampling was also incorporated. So if there were any livestock in the area that were presenting ticks, they were taken. Also, if there were just any present on the ground in the surrounding area, they were obtained also. And during sample collection, environmental data on the temperature, humidity, land cover type. I think, was that everything? There was some more, weren't there? Soil moisture. They were all obtained as well. And this field work was planned to be completed in 2024, but it had to be cut short because there was a drought at the time of sampling. So it was then repeated in 2025, but I believe there might have been flooding during sampling as well. So 2 weather extremes there for our samples. So once these ticks were collected, they were sent over to the University of Liverpool, where I have been identifying them using morphological keys. And I've also been extracting DNA from a single tick leg to preserve as much as the specimen as I can and all of the internal microbiome. And we've sent the DNA extracted from the tick leg off for Sanger sequencing so we can validate my morphological IDs with the genetic data. But I think we need to incorporate a few more molecular markers because the one we've chosen has come back with some dubious species assignments. So that's an ongoing process. And I've also been optimizing our PCR to recover protozoan DNA and bacteria DNA. And these are using universal primers. So we want to target all the protozoa communities within the ticks and all of the bacteria so that we're getting the pathogens and the commensal microorganisms, and we can get a really good idea of the community structure from within the ticks. So here are the results from this survey. So overall, 853 exodied ticks were collected. 82 of those were collected with the blanket sampling method from the environment. There were 771 opportunistically collected ticks, and these pertain to 4 genera. So the most prevalent of these were the hyaloma ticks, 49 collected with blanket sampling and 361 collected opportunistically. Second most prevalent tick were the Rhipicephalus species, followed by amblyoma, and lastly Haemaphysalis. There was only one of these ticks from this genus in the whole of the subset. So here is a map showing pie charts of the distributions of the different genera across locations. It's quite nice to see how the genera are kind of separate and localized, with the amblyoma, which was just one sample collected from blanket sampling. And then we've got the opportunistic pie charts. So we found far more ticks with this method. And you can clearly see the amblyoma localized to these areas here, which coincide with previous maps that we've seen today. I'll also just mention as well that out of the 86 locations where blanket sampling was conducted, 56 of those returned no tick species from the environment. And this is also a really important finding because we can then input the environmental data collected from these locations and use it to inform our habitat, the tick habitat preference models. So it's still a really important finding. So these are the breakdowns of the different species within each genus and where they were found. The blanket sample ticks are those represented by the diamond shape and the points of the opportunistic ticks. So by far the most common species that we found were Hyaloma truncatum ticks. They were in so many of the samples. We also found Hyaloma rufipes and Hyaloma tyrannicum, which Virgil, I noticed you found like the same 3. And then the Rhipicephalus species. So lots of different, a very diverse genus within our subset. With this one, it's really interesting because this was the most prevalent tick genus from the blanket sampled consortia, and a lot of them were in their nymphal stages as well. And I found this really interesting while I was identifying those because some of them had these really unusual adenal plates, which had like the 2 prongs. I've tried to include a picture down there. I couldn't actually find much online or in the morphological keys I was using about this, but my guess was tricuspis. So hopefully we'll be able to get this validated with Sanger sequencing. And next, amblyoma. We know that this is the causative vector of heart water, so really important. And we found, yeah, there were very distinct ranges of these two. And lastly, haemaphysalis, just the one from the blanket drags, and that existed in the centre of the map there. So next steps. So as Hannah mentioned, we're sending off the first batch of our DNA extractions next week. So what I've been doing is, after morphologically identifying them, I've been cutting them up and extracting DNA from the whole tick. And I've been validating the presence of bacteria and protozoa from the samples. So we're going to send these off for Illumina sequencing at the Centre of Genomics Research in Liverpool. And what we're hoping to find are communities of protozoa and bacteria within each tick. So we can have a look at the commensals and the pathogens that are coexisting together, and how these correlate with any environmental factors. And these results will then be channeled into a model, which is being worked on by our postdoc, Iddon. And it's this comprehensive results that will help to hopefully inform future tick-borne pathogen transmission control. So what we're hoping to find is something that one of Hannah's PhD students has done previously, whereby they characterize the nematodes communities of wild animals using Illumina sequencing. So they were able to genetically identify all of the different nematodes from their fecal samples. And these are just the acknowledgements for everyone on our project. Thank you for listening.